All right. Can you please confirm if my screen is visible?
Yes, the screen is visible.
Okay, thank you.
And that might be a content.
All right. I can see that few participants have joined. We'll be starting with the webinar in just few more minutes. And meanwhile, we are waiting. It would be great if the participants can introduce themselves in the chat box. You can let me know about your education background and research interest. All right, so without further delay, let's start with today's webinar. So, hello everyone, a very warm greeting to one and all. My name is Sridevi Krishnakumar, and I would like to welcome you all for the webinar on OmicsLogic, Bioinformatics, and Data Science Research Fellowship Program. The Research Fellowship Program has been designed to help young researchers and students take advantage of the bioinformatics resources for the analysis of complex life science data and become well versed in bioinformatics. Research fellows participate in cutting-edge bioinformatics research led by expert mentors from academia and industry. The fellowship program will offer a combination of online resources and mentor guidance to prepare you and help you complete a bioinformatics project. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of our mission, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by over 24,000 participants from 187 countries in over 300 workshops in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, and data science for biomedical data. And as a part of the training program, you will have access to the OmicsLogic resources, which takes a combination of training modules, data analysis tools, curated project data sets, and interactive sessions with mentors to give the student a clear path of the Bloom's taxonomy pyramid. And this also comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. The OmicsLogic platform also gives options for analyzing a given dataset, and these datasets and associated publications can be used in several ways. First, you can learn bioinformatics without any coding at all. For those interested in opting the non-code pathway, they will be given directions to access the tBioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipeline. And different stages of analysis are performed in different sections on this multi-omics platform. For those interested in learning to analyze data through coding, we provide the OmicsLogic Code Playground, which will help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in biological data. So now to view the various courses and example projects and student projects, you need to first sign up on the OmicsLogic Learn portal. So for that, let me walk you through on how you can sign up. So here is the link for signing up. I'm pasting it in the chat box for the participants. Please visit the link and I'll walk you through how you can sign up. So when you visit the link, you have the option to create an account. For those participants who have not created an account on the portal yet, for them, please click on the create an account option that you see here. And once you click on this, you can enter your name, provide an email ID and password, and you can sign up. Or you can also sign up using any of your social media handles. This could be your Google, Facebook account, Apple account, GitHub account, or even Twitter. And for those participants who already have an existing account on the portal, you can simply click on the login now option that you see here and simply enter the existing email ID and password and click on sign. So with that, I'll pause for a couple of seconds for the participants to sign up. And once signed up, please put a yes in the chat box so that I know I can proceed ahead. And if you already have an account on the portal, then please put a done in the chat box as well. All right, Walter, were you able to sign up, or do you have any queries on signing up? If so, I'll be happy to explain once more. And along the way, if you're facing any technical issues while signing up, you can let us know as well. And our team is here. They will be happy to look into it and get it resolved. All right, so without further delay, I'm just going to click on sign in. But please let me know in the chat once you have signed up. I'll be monitoring the chat as well, side by side. So since I already have an account on the portal, I'm just going to click on sign in. So once you sign in, you will be able to see the various courses that are there on the portal. So once you scroll down below, you will see a search bar that says looking for a specific course. So here, after you sign up, you can enter a keyword that you're interested to learn more about. So let's say you're interested in transcriptomics, then you can enter the keyword transcriptomics, and that will show you the various courses and example projects that make use of the concept on transcriptomics. So as you can see, there are several courses on transcriptomics. One is transcriptomics, and one is on single-cell transcriptomics, and then transcriptomics using R programming and Python programming, and several example projects, such as post-response to malaria infection, and several other example projects that you can take a look at your own pace. So this was what you can see under the courses tab. So for now, it is important that you have to set up your account. For that, you can simply click on the blue bar that is welcome back with your email ID. So once you click here, it will take you to your account settings. So it is very important that you have a completed profile on the portal. To complete your profile, you can simply click on the icon that says profile. And this will enable you to edit the features on the portal. That is, make sure that your name is in proper capitalization, and that you add a brief bio about yourself that will consist of your education background and research interest, and add a profile image for yourself as well. And also link your social media handle to the portal. So once you update your profile on the portal, it will show 100% complete. And then you can also take a look at the various tabs that are there on the portal. So for instance, under the activity tab, the mentor of the program and the community manager will be tracking your learning progress. Under the courses tab, you will be able to see the various courses and example projects that you are currently taking. Under the projects tab, once you complete a project as a part of the program and publish it on our portal, that will be displayed under the project tab. Under the Programs tab, once you enroll for the Research Fellowship Program, that will be displayed under the Programs tab. And finally, after the completion of your coursework with the required assignments and quizzes, the certificates will appear under the Certificates tab. So that was all about setting up your profile and the various tabs that you see here. And another important tab that I want to show you is the Projects tab here. Here, once you click on this, you will be able to see the various student projects that have been carried out as a part of the Research Fellowship Program. And these projects topics range from oncology, infectious diseases, precision medicine, machine learning, neuroscience, and so on. So the participants can have a look at all of these student projects that are there on the portal. And for today's webinar, we are joined by one of our research fellows, Michaela Shetty, who will be presenting her research project that has been carried out on Alzheimer's disease. So with that, I'll pass on the stage to Michaela. The stage is all yours.
Hi everyone. Hi Srigori.
Yes. Hello, Michaela.
Srigori, is it possible for you to present my slides from your side, please?
Let me take a look at it.
Sure.
Okay, is my screen visible?
Yes, it is. Thank you so much.
All right.
Okay, so hi, good afternoon, everyone. Good evening in your respective time zones. My name is Michaela Chetty, and I have been working on transcriptomic analysis of human beta amyloid in the model organism, which is a nematode, the C. elegans. So that is my project topic that I have worked on with OmicsLogic. But before I go on to that, let me just introduce myself and say a bit about myself. My name is Michaela Chetty. I have completed my BSc in biotechnology and my MSc in zoology. After that, I took a gap year. I worked on some research projects, and as we all know, COVID hit. Yes. So that's when I started developing more computer skills. I am currently doing research at an institute, and I also work on C. elegans, but in the wet lab. So C. elegans has been one of my favorite organisms because of its close similarity and its, evolutionary companionship that it had with humans around 600 million years ago. And that's why they are good to work with. The other very important factor to take note and into consideration is that when we extract human samples, they might not always be in the best condition possible. For example, if we're talking about neurodegeneration, then extracting brain samples or, DNA or proteins from the brain itself, it needs to be maintained in the proper sterile environment, in the proper temperature, in the proper atmosphere that is suitable for that sample. Whereas if you're using a model organism, I could say that research is a little bit more friendlier or easier, if I could put it that way. Which C. elegans is grown in a BOD 22 degrees on an agar plate. So not much stress and fuss is over there. My interest lies in neurodegeneration primarily and Alzheimer's disease. I have been affiliated or, attracted towards Alzheimer's disease for, I would say, the past 10 years. It's a bit of a personal reason, but it led into a scientific affiliation after I started my bachelor degree. And through neurodegeneration, I have planned or, intended to further my career. And I came across OmicsLogic, their research fellowships online. And I thought, wow, this is a great opportunity for me to, get on some hands-on skills. And it's definitely going to help develop my profile. And I'm going to learn new skills that I can add to my CV. I can use the skills to further analyze more data. And while analyzing more data, you get to know more of the proteins or more of the gene sequences or more of the comparative analysis that can be done. So that's a bit about me. And I have started with the OmicsLogic portal by doing a few of the basic courses because I am not originally from a bioinformatics background. I'm from a biotechnology background. We did have the course, but it was a subject. So I started with Introduction to Bioinformatics. I further did the Bytes and Molecules course that is available on their online website. And I further did the transcriptomic course, and I developed more skills and interests and questions from, these three main branches of courses on the OmicsLogic portal. So without further ado, I will continue on to talking about my project that I have done. So on the screen, I have listed, my objectives of the project that I set out to do. And my data was available online through NCBI. And I had taken these three objectives based on the data. And like I said before, my filtering method or my choice of data was done by my interests. So my interest is neurodegeneration and C. elegans. So when I had put this into GEO online, I had found that they had only given me around 7 or 11 data samples. And obviously I put the C. elegans filter, and that's how I got such a smaller number of data available online. And this one, this data set had attracted me a little bit more than the rest because of its vast number of samples, and they had used a C. elegans strain that represents amyloid beta deterioration or neurodegeneration. So that was really interesting for me to see the computational biology aspect of it, because we know that the transgenic C. elegans is available, but when you get its data, it's a different thing because you can further conclude more results. That is how I chose my data, and the objectives that I have chosen are listed on the screen. So my first objective was to see the changes in human beta amyloid peptide gene expression. And the second objective was to identify the pathways that were taken or triggered by amyloid beta toxicity over time. There were different time points. And the third objective, one of the prominent ones, I would say, or important, because that gives it its human significance, was to see the orthologous biological functions in humans. So as we all know about neurodegenerative diseases, they are coming to be quite common these days, I would say. Not necessarily a good thing, because it leads to a downfall on one's life. And that one being normally an elderly person, generally above 50 years of age. So in the brain of a person who is diagnosed with Alzheimer's disease, there is a specific protein called the beta amyloid protein, which is involved in Alzheimer's. I must say that the beta amyloid protein is one of the accumulants, or it has been said that when excessive accumulation of amyloid beta, it causes Alzheimer's. And that's why there is studies done on the protein called beta amyloid or amyloid beta. It's pretty much interchangeable. Based on your lab or your location, different people use different first words for them. So the beta amyloid protein, which is involved in Alzheimer's, it has different molecular forms that it takes place between neurons. As you can see in the picture, it's on the end of the part of the neuron. So it is formed by a larger protein, which is broken down, and the larger protein is called the amyloid precursor protein. This is normally the beta amyloid 1 to 42, as it's commonly known, and it is toxic. What happens is that the proteins clump together. When they clump together, they form plaques. And these plaques collect between the neurons, and they do not allow the cell to function appropriately or function in its rightful manner. So that is the purpose of studying beta amyloids. And just on a side note, the tau neurofibrillary tangles, it's also known as hyperphosphorylated tau tangles, the chemical structure of it. So it's inside the neurons. So healthy neurons are supported by internal structures known as microtubules. And these microtubules are very important in guiding nutrients and molecules from cell bodies to axon to dendrites. But when tau tangles are not in proper form, or they are sticking to other tau molecules, they form threads, and these threads eventually join together to form tangles inside the neurons. What happens is that these tau tangles or hyperphosphorylated tau tangles, they block the neuron's transport system, and in, you can say, long-term effect, this harms the synaptic communication between neurons. So this is just a bit about the molecular and basic molecular and biochemical aspect of amyloid beta and tau tangles that happens. And what happens is that in some cases, or in most cases, the dead and the dying nerve cells, because they're not getting proper nutrients, they end up getting twisted and tangled between each other. And beta amyloid, the chemical form of beta amyloid, gets sticky. And when it gets sticky, they group together because of sticky ends. When they stick together, they form plaques, and it blocks the signal synapses. Ideally, not good for the human, or for anybody, because that's the basis of neurodegeneration. And you can say it's an aiding factor to neurodegeneration. There are additional factors such as genetic and environmental, but this is one of the most prominent ones, and literature shows this quite evidently. So going on to my project, we had taken a sample or the data set that is available online, and I have listed the PMID over here on the screen. In the data set, there were 3 strains of C. elegans. So each strain contained a different I could say a physical feature or the biochemical feature. So in worm strain CL4176, this had the human amyloid beta. In worm strain CL2337, this carried the protein expression or neurodegeneration of amyloid beta expression. So this had, the amyloid beta plaques, basically what is trying to say. And worm strain CL2179, this was used as a negative control to evaluate and see the results that we were getting, as a comparison method. So why do many labs use C. elegans? Apart from it being a model organism, C. elegans is back, many years ago, like I mentioned before, 600 million years ago, there was an evolutionary theory split that took place. They had a common ancestor with humans, and the common ancestor was known as Urbitellarian ancestor. So the split took place, and that's how the nematode C. elegans, commonly known as a worm, looks a little bit different. Okay, not a little bit, quite different to a human. But you should know that they have a minimum of 60% genes that are homologous. So that's quite a large number to note down, despite the physical or phenotypic differences that have been noted. So C. elegans are hermaphrodite, which means that they have 2 sexes in them. They have sperm and oocyte. First, the sperm is produced, and then the oocyte passes into the spermatotheca, and then it's fertilized. That's how fertilization takes place in a hermaphrodite. They grow and multiply in large numbers. So in a laboratory, one does not have shortage of sample or shortage of organism. That is not very common in a C. elegans laboratory. It is grown on a NGM agar plate or a Petri dish. As you can see on the bottom left on the screen, there is an image of a Petri dish. So liquid media is made and allowed to solidify, and they are grown on the media. They have a very fast life cycle. It usually takes around 24 hours to get gravid worms again, which is quite a good time span for experiments. They do allow for long-term cryopreservation, which means that you are allowed to freeze them if in case COVID happens again, then, you know, our samples will always be safe and stored securely. Going on to the practical experience that I gained through OmicsLogic, I extracted the data from my PMID that I have listed above, and I filtered it and sorted it out in terms of time frame. I then performed the t-test. So the t-test is essentially quite important, I would say, because in a statistical means, it's a measure of probability that you can obtain your result from. So you can see the difference between groups. In my case, I had different time groups. I had different time frames. Time frame for a 12-16, and this was done. So ideally, lower the p-value, the more confident you can be that, you know, your observed effect is real. And this is in terms of when you are using grouped data, which in my case it was. And after that, I did the p-adjusted value, which gave me more statistical data that I could use to filter out and see my data. I then, after the p-adjusted value, I then ordered it in terms of number-wise. So I could see which was going to be upregulated and which was going to be downregulated, which gave me a good idea. And, you know, so when you get your p-value, you can see the numbers. Some of them come in negatives and some of them come in positives. And yeah, so just give me 1 minute. I think the internet is. Hello, can you hear me?
Yes, we can hear you.
Okay, sure. Okay, so after doing the t-test, and then we did the p-value sorted, and after that we did the p-adjusted, and after that the log fortune. So this was pretty much the process of and the method that was taking place. The differential gene expression analysis was significant because we use 95% of confidence, which means that we use 0.05, and this gave us a good evaluation for our data. In my case, the data set of time frame 16 was very significant. I used this data to get a Venn diagram, which is like a comparison graph, or, you know, a chart that you can say, where I've put all my data into, and time point 16 was most significant in the sense that it had more number of values that needed to be considered. So after doing this, I further went on to the gene enrichment analysis, which there are many sites available for the model organism, C. elegans. So I used Wormcat to get my data, and I used another website called AuthorList. So AuthorList has quite a number, I mean, more than 7,000 protein coding genes of C. elegans itself. And it gives us the human ortholog to that specific, you know, protein. So on the screen right now, you can see the data that was resulted from upregulation. And this was done through Wormcat, like I mentioned. Wormcat is an online website that anyone can access. There's no restrictions. Yeah. So my data received that the data that I got was quite, I would say, normal, or I would say me knowing about neurodegeneration, it was a little bit good because microtubules are very common in neurodegeneration, signaling phosphatase and transmembrane proteins, collagen. These are all very common things that take place in a neurodegenerative organism or human. But to my surprise, there was a very uncommon, or two uncommon things, I would say, and they were amongst the top 4. So I think anything in the top 5 is very significant and relevant in any data sets. So number 1 being regulated by multiple stresses. And when I further went to evaluate this in a biological manner, then I saw which genes specifically, it was mostly the DAF genes, DAF2, DAF12, DAF16, that were regulating multiple stresses and what type of stress regulation did they cause. The other surprise, if I would call it, is major sperm protein. And I would normally not assume major sperm protein to be in the neurons. But I think, major sperm protein could have some kind of immune response significance or symbolization or structural similarity. And hence, because of, the protein fold and the immunoglobulin structures, there might be some kind of similarity in that. And that's why I think computational biology is very important, because it shows us a different aspect, or it gives us a different pathway that we can take to explore what we already know, or what we might not know for that matter. For example, there has been studies done on mice and the immunological effect in neurodegeneration. So there might be not any studies done on humans, but because of bioinformatics and computational biology, we can use this data and translate it into medicine or into wet lab, where we can further develop or ascertain the cause or the treatment plan for neurodegenerative diseases. So on the next slide, I have just elaborated a little bit about microtubules. For those of you who are new to the neurodegeneration field, microtubules are very important, and they are there in the structural network. So they are, like I mentioned in the beginning about the tau tangles, so it belongs to a kinase superfamily. And there was a specific ortholog of human, TTBK5, that was very significant in my readings. This I have mentioned here as well. So it can be taken down, into further analysis. But I mean, we first needed to process the data to get this kind of outputs. Another one that was significant on the next slide was the transmembrane protein, which is also very crucial for cell membrane, because in neurodegeneration, that's what deteriorates a lot faster, like, APP, amyloid precursor protein, and hypophosphorylated tau protein. So these tau and amyloids, they were enriched or, affecting the dopamine neurons and in any other musculature and nervous structures that were there. But it can also be further studied. I feel in science, there's no dead end. Last but not least, my most prominent one, I would say, is major sperm protein. And this definitely needs to be examined further. But we have seen, and a lot of number of genes have come out of the major sperm protein family. So this is something that needs more studying. Maybe its immunological structure is similar, or something to do with the immune response cells. It can be related to anything, in relation to that, and that will give us a better idea. So overall, this is a basic idea about my project that I have done in neurodegenerative diseases. And I am further working to put this together in an informed manner for a publication. And if given the chance, then I would, definitely take up the MSP at further studying levels, or, any of my other significant readings, such as the DAF gene that have been seen in stress-regulated, the first bracket of multiple stresses. So there were a lot of DAF2, DAF12, DAF16. So this can be further studied in terms of lifespan, aging, and longevity. But as of now, I am working towards a publication, and that is the direction that I am going in. And I definitely learned a lot through the project. And thank you for having a wonderful team that supported me.
Thank you so much, Michaela, for that wonderful presentation. And before we wrap up the presentation, I just have one quick query for you. So what advice do you have for those students who want to get involved in research, but they do not know how to get started? What advice do you have for students?
I would tell you to identify, firstly, what suits you. As a biologist myself, are you a wet lab person? Are you a dry lab person? Okay, so by wet lab and dry lab, I mean dry lab being the computer stuff, RNA sequencing, PCR data, transcriptomic analysis, molecular docking, cheminformatics. That's the dry lab stuff. If you are a wet lab person, that means you enjoy being in the lab, working for long hours, very long hours. Micropipette samples, assays, proteins, fluorescence, spectrophotometer, mass spec, that kind of stuff is wet lab. But at the same time, while you are identifying your niche or your category of what is for you, I would advise you to gain some skills and techniques through online methods, be it learning a program language, R or Python. Or if you're interested in proteins, go for molecular docking. If you're interested in genes, there's genomic biotechnology. If you're interested in transcriptomics, there's RNA, there's microarray data, there's RNA sequencing, single cell. And because the wet lab and the dry lab, I believe, I believe as Michaela, they work hand in hand. So you can always count on one another to double check or to verify or to add to your readings or your findings. So for somebody who's new, I would tell you to read a lot, to explore your options, to gain skills, and to definitely not to continue learning, don't stop. You find your niche, whether it's DNA or proteins or mass spec, metabolites, metabolomics, ligands. There's so much of things, and a lot of this can be done online, in the dry lab as well. So in the meantime, if you're not getting any opportunities, or if you're still shooting your shot, trying to send out applications, everybody has a laptop these days. It's very common. So just try to work on a project online, or through online means, or dry lab stuff is what I'm trying to say, in the meantime of you getting your wet lab project going. But I believe that they work hand in hand, and that's something that I strive to do for my further projects.
Perfect. Thank you so much for that response. And one final query. I see that you have enrolled for another program on cheminformatics. Do you plan on continuing in the same field using cheminformatics, or is it a completely different purpose?
So my passion is neurodegeneration, and I will be honest to say that I'm staying in neurodegeneration. But I love proteins, and they fold in very meticulous and fancy ways that I need to know more about. Hence my enrollment for the other program.
Thank you so much, Michaela, for joining with us. And that was a really wonderful presentation. And we look forward to seeing more about you from in the upcoming days about your research publication.
Thank you.
And with that, now I'd like to pass on the stage to one of my colleagues, Sonalika, who will be explaining more about the research fellowship program and the various specializations. Over to you, sir.
Thank you so much, Sringari, and thank you, Michaela, for such a wonderful presentation. And I would like to discuss about the program structure, outcomes of the program, and also I'll take up your queries and listen to your questions and answer them. And before I do that, I want to know in the chat box who is our audience. So I just wanted to know if you all are students, graduate or postgraduate students, or PhD scholars or researchers from bioinformatics field or non-bioinformatics field. Please put in the chat. Yes, Usma, we have recorded this. Please let me know in the chat what is your background and what is your research interest so that we could have this interaction based on the field that you come from. I'll be awaiting for responses, and meanwhile, I'll just begin ahead. So we have been discussing about this research fellowship training that has been designed to help young researchers and students take advantage of bioinformatics resources for the analysis of complex life science data and become well-versed in bioinformatics. The research fellows generally participate in this cutting-edge bioinformatics research projects that are led by expert mentors, and the fellowship program offers a combination of the online resources that I want to show you today and the mentor guidance that prepares you and helps you complete a high-quality computational research project that could be either submitted as a dissertation work in your college, or it could also be published as a part of your thesis. So we have been discussing a lot about bioinformatics, data science, different types of big data being generated. We know that this field is constantly growing and developing new tech and software to improve processing time and power. The data sets are generally analyzed are very large, and they take immense amounts of computing power and memory that many personal computers do not generally have. Also, despite being publicly available, the data is so complex to access, and also coding knowledge is usually required to do so. Similarly, the analysis pipelines and the programs, they require coding knowledge that many biologists do not have. And in a university setting, the students, they generally have the opportunity to just learn the background information of designing and implementing a research project, but they do not receive the practical hands-on experience that I should say with the full workflow due to the limitations that I just mentioned previously. Similarly, faculty also face challenges with being able to cover a diverse range of topics if they're specialized in a certain area. Let's say if they're unfamiliar with the new tech in general, or for example, a professor whose research focuses on oncogene expression might not know much about metagenomics of seawater or neurosciences, as Michaela mentioned. So to dwell with this specific issues that we come up with our general academics, or with regards to the computational requirements that are required to work with big data, we have this research fellowship program where we offer resources that you can utilize to get yourself up with the skill set that is required to work with these different types of data. And also you have a platform where you could actually analyze your big data that comes from different types of omics data. So we aim to utilize this beginner level to advanced level project-based coursework and the case studies that are published on the portal that generally focuses on data science techniques and programming languages and machine learning methods. So these techniques and different programming languages could be used to analyze these different types of omics data. And by omics, I am collectively speaking about genomics, transcriptomics, metagenomics, epigenomics, and also single-cell transcriptomics. Throughout my talk, I would be discussing a lot about this omics term. So I'm by omics, I am referring to all of the studies collectively. So this research fellowship program would include a high-grade training material that I'll just take you through, where we'll discuss about these different omics techniques. Now, let's say once we are well equipped with these omics techniques, There are various research specialization tracks in which a research fellow, a researcher, or a student can actually work in. So we get these questions by students from different universities and degrees, where they ask where they could apply a specific type of skill set, in which research track should they be working on, or which research field they should pick to carry out a certain type of interest that they have. So I want to discuss all of this today and help you know about these different resources that you can leverage. So this is the OmicsLogic Learn portal where Sri Gauri had asked you to sign up, as you have a bunch of courses that you get an access to as a part of the research fellowship program, which includes different omics and data science coursework. And all of this is in regards to biomedical data analysis only. So as a student or a researcher who comes from a different background, let's say non-bioinformatics background entirely, a microbiologist or a biochemist or medical sciences, and you're interested to transition into this field of bioinformatics, we get started with the very basics of bioinformatics through a course that is Introduction to Bioinformatics. So right now I'm explaining the program structure to you. There are two or three rather types of program structures that you can enroll yourself into, either 2 months or for 3 months or for 6 months, based on the time you have, or I must say the dissertation period that your university has asked you to complete in, or your research proposal if you're a researcher working with a lab. All right. So if you come from a non-bioinformatics background, you get started with a course on Introduction to Bioinformatics. This course includes basics about what is bioinformatics, what is the role of big data in bioinformatics, what is the role of bioinformatics in healthcare, and what is translational bioinformatics, discussing the applications of the field bioinformatics in various research specialization tracks. Now, if I talk about what is inside these lessons, we have textual concepts, videos are there, images are there, research publications are there to supplement your knowledge with the same. And towards the end, you have some quizzes and assignments put together to access your knowledge that you gain from a specific lesson. Now, when we discuss about different omics fields and different research specialization tracks, as a beginner, you might be unaware of what these different omics fields are, or what these different specialization tracks are that you could really work upon. So we have put together a course that is Bytes and Molecules that you can go through if you are a beginner and you want to learn about these different types of omics fields. Many of the students who join us, they have a question that they want to do a project in the field of bioinformatics, but they're not sure which type of data should they work with, or which type of field should they apply a specific type of data analysis that they would learn. So for these students, we take them through this process where we teach them about all the different types of fields, an introduction, obviously. And based on their interest, then we take them a step ahead and take them through a specialized training process. Now, let's say if a research fellow joins, goes through the Bytes and the Molecules coursework and says that I'm interested in transcriptomic studies. So the next step for them would be to take up a specialized training in transcriptomic studies. Now, what do I mean by a specialized training? If it's transcriptomics for a student, then it is important for them to know what type of sequencing techniques are there to sequence transcriptomic data, what type of data format is generated from these sequencing techniques, how to analyze the different types of data formats that come up from these techniques, what are the algorithms that could be used, what are the steps of pre-processing, mapping, quantification, and then carrying out the downstream analysis for transcriptomic data. And in terms of algorithms, what do they do? What type of parameters they take? What type of outputs they give you? And then how do you interpret them? So there is a flow from data generation to biological interpretation. So that flow is taken up in this specific coursework that a research fellow has to then go through and get a good hands-on understanding with transcriptomic data. In all of these courses, you'll find an example data set that we have taken up with which we are working. And for practice, you can take up the same data set, practice the pipelines. And by pipelines, I am referring to a platform that is the Bioinfo platform. So we have two platforms for us. Those who are new and are joining us for the first time, this is for them. One is the OmicsLogic Learn portal, where you learn all of these different types of fields and research specialization tracks. And then you have the Bioinfo platform, where you actually carry out the hands-on analysis or practice these different types of pipelines. Now, there are two types of people who like to code, who want to learn how to code, and the ones who don't like to code, or they are just code biologists interested in the insights of their results, but do not have a good hands-on with coding languages. So for those type of people, the best platform is the Bioinfo platform. And also, if you are a coding person, you definitely want to process your data from FASTQ files to get a gene expression table at least, which requires a high computing system. Otherwise, if you don't have the platform. So in that regards, the platform has some AI built machine learning tools already put together. And these pre-built tools could be used to process your data, map the reads on the reference genome, quantify the data, carry out downstream analysis, do the two different types of processes at different steps. I won't go much in depth of the platform right now, because my aim today is to discuss about the resources and the program structure and also the research specialization tracks. Okay, so now when I talk about these different pipelines, how do you use them? What type of results do you get? Everything is there in the lessons itself, in the course that you take up. Now, once your course is complete, the next step comes to submit a research proposal, where you put together what is your research interest, in which field you want to carry out this research. You have to explore various public repositories to look for a data set that you would like to work on, and submit that data set link as well in the proposal form. Throughout this program, in the beginning in your training session, you would be also taught how to explore various public repositories, how to extract the data, and how to prepare it for further analysis. Now, where I talk about different research specialization tracks, I want to mention here what are those tracks on which we specifically work on. The first one is infectious diseases. We have put together this page on research specialization tracks and topics. I'll put the link in the chat box. On this page, we have put together the research specialization tracks and the type of data analysis that is possible under each one of these different specialization tracks that you can work on as a research fellow. One is infectious diseases, and then you have the list of different data analysis that is possible under this field. Then you have oncology, and also on the page itself, you have some example projects or case studies that we have worked upon. They're also mentioned on the OmicsLogic Learn portal. I will be just taking up these points very briefly here, keeping the time limit. And you have the link, we'll share the link with you as well. You can take your time and explore these different topics that you would like to work on when you join the program. The second field is oncology that you can work on with applications or with the usage of different types of omics data again. And also multi-omics integration is possible too, and structural bio and cheminformatics work could be done as well. The next type of research specialization track you have is precision medicine. And under precision medicine, again, different types of data could be analyzed, like transcriptomic data, clinical data, epigenomic data, and genomic data. The next field is agriculture. Under agriculture, we have an example project on potato drought resistance. So those who are interested and are curious to learn how an agriculture-based study could be done using these different types of data, could go through the portal, go through this case study, and you have these research questions that we have put together for them. The next field is neurosciences, and then metabolic disorders and space omics. We have put together some research questions for these different fields as well that a researcher could work upon, and different types of analysis that is possible. So in total, there are 6 fields that I went through: infectious diseases, oncology, neurosciences, agriculture, precision medicine, metabolic disorders, and space omics. Now, if you are interested to work or take up any one of these different research specialization tracks and are curious what type of analysis could be carried out, you're free to go through this entire page. And if you have any questions, you can get back to us on our email IDs, or we also have a WhatsApp group. We can share the group link with you, and we can meet and discuss your research interest and get you started with your journey of the research fellowship program. So that was it that I wanted to discuss. And now I would like to pass on the stage to Shri Gauri. Over to you, Shri Gauri.
Thank you so much, Madhika. Let me share my screen and discuss about what are some of the research fellows been up to after the completion of the program. So you might be wondering what comes after completing the courses and example projects as a part of the program. So the answer is possibilities are endless. So to begin with, majority of the students are focused on publishing their work. For instance, Rupri Vaja, a biomedical science graduate student, she focused on assessing the underlying reason for the development of gray matter lesions in multiple sclerosis patients at their transcriptomics. And further, she went on to publish her work in immunogenetics, an open access journal that focuses on the genetic research areas, autoimmune disorders such as multiple sclerosis, rheumatoid arthritis. And another interesting student publication is that of Shupanta De, a genetic engineering student. He studied the effect of malaria vaccine on the transcription profile of patients to determine its efficacy and understand the molecular mechanisms underlying the protection that the vaccine provides. He also went on to publish his work in Genomics and Informatics, which is the official journal of the Korea Genome Organization. Presently, like them, we have several other students who are working on their publication, just like Nivedita Arambam, a biotechnology graduate student. She's working on transcriptomics study of cancer treatment on patients and its after effects leading to relapse. And like her, we're looking forward to several other publications from our present students. Further, you also have the opportunity to participate in Omics Research Symposium and poster presentation competitions to win exciting cash prizes. And this symposium is an international symposium that brings together experts such as Dr. Gareth Trabul, staff scientist at Lawrence Livermore National Laboratory, and Dr. Joseph Borg, associate professor at University of Malta, amongst so many other experts who join as guest speakers and judges. And the student projects are not just limited to graduates and undergraduates. For instance, Andre Modeno Sosa, a high school student at Mexico, presented his neuroscience project at the Regeneron International Science and Engineering Fair, for which he won the Potamkin Prize and the 4th award in the biomedical and health sciences category. And this program can also be integrated as a part of the existing university curriculum. For instance, we collaborated with the Systems Medicine graduate program at Georgetown University, where the lecturers were prepared considering the interest of the program director to address challenges relevant to clinical application and biomedical research. And the training ended with students submitting their research project on the portal. And the topics range from, as displayed in the screen, from infectious diseases, metabolic disorders, neuroscience, psychiatric disorder, oncology, machine learning, and so on. So all of these projects have been carried out on machine learning using Python programming. And these projects can also be showcased for higher studies and international university applications, like some of our research fellows, like Dilara Raikin, who got accepted to the Erasmus IMBRC program in marine biology, and Dhruv Mehra, who got accepted to the Precision Medicine graduate program in Manchester, and Anusha, who got accepted to the PhD program in metagenomics at the University of Sweden. And faculties and researchers have also collaborated through our programs to publish their research work. And our latest collaboration has been with a team of researchers at Noguchi Memorial Institute for Medical Research at the University of Ghana. They carried out to understand a computational study to understand the variants associated with sulfadoxine resistance in Plasmodium falciparum. And it is also possible to pursue an entrepreneurship and business innovation track like a team of industry researchers from Biologic Support Services, a biotechnology company in Nigeria. Their aim was to improve the fertility of women in Nigeria and improve their chances of carrying their babies to term by developing a test kit. And this entrepreneurship track is not only pursued by researchers, but also by students. For instance, a group of undergraduate and postgraduate students from Vellore Institute of Technology worked on developing an early detection kit for the detection of neurological disorders with a specific focus in Alzheimer's disease. So these are some of the research outcomes that I wanted to showcase. So now to get enrolled for the program and to understand what are the various registration options that we have, I'd like to pass on the stage to Shubham Kumar, who will be explaining more on. Over to you, Shubham.
Thank you so much, Srigori. And hello, everyone. I'm Shubham. I'll be like briefing you about the registration part for the Bioinformatics and Research Fellowship Program. So, Shigori, could you confirm if my screen is visible?
It is, yes, now we can see the program page.
All right. So as you all can see that this is our the program page, or you can say the landing page also. So here you will get to know about the all the informations like what all you will be getting or why we have designed this process. So as you can see, this program has been designed for the to help the young researchers and like students who are working on their dissertation project or for the thesis work, so that they can take the advantage of bioinformatics resources and work on the complex data analysis part. So what we all will be providing is like you will be get trained, along with that you will get the mentorship from industry academy experts. And apart from that, as Sonalika mentioned, you will be getting the access of T-Info server, which is a cloud-based platform for big data analysis. So as we scroll down, you will find that we have like we can say three-step process for our research fellowship. So first you will be getting, let's say you are a beginner for the bioinformatics field. So as earlier mentioned, you will be like providing with some of the trainings from our OmicsLogic Learn platform, where you will be going through the courses related to your field of interest. And while going through the courses, let's say you come up with a research topic or a research question to work on, so that will shift to your second step. That is, you will be working on your research topic, as well as you will be working on your literature review part, so that you come up with the best research question and proper data set to work on your analysis part. So in these steps, you will be working on the second step, you will be working on your data sets and the analysis part. And after it is completed, you will be like shifted to the third part, which is the presentation of analysis for the expert feedback and the publication part. So as we will scroll down, you can see here that we have three different tenures, or you can like four different tenures, like 2 months, 3 months, or 6 months. So the common tenures which we have for all these tenures is you will be like going through some of the asynchronous courses, and apart from that, you will be working on some of the hands-on assignments or hands-on projects also, and group sessions will also be there. And let's say if you miss some of the sessions, then we will provide you with the recordings also. You will be getting the hands-on coding sessions, and apart from that, online technical and mentor support will always be there. And you will be also getting the access of the OmicsLogic program access and the educational cloud pipeline license for your T-Info server. Now the extra benefit what you will be getting for the 60 days tenure is like you will be like getting a chance for the literature review part. And after that, you can come up with a proper research project report work. And as mentioned by Sri Gauri, you can like showcase your research proposal in a form of a poster at various international symposiums. Now coming for the 90 days part, all these like the outcomes you will be getting for the 90 days is like literature review, then you will be getting the one-on-one mentorship guidance, and after your project completion you will be getting a chance to for your publication part. So on this page, when you will scroll down, like you can see like some of some of the projects by our previous research fellows related to the infectious diseases. Then in the field of spaceomics, they have performed some of the project. Then in the field of precision oncology, then machine learning, skin diseases, and data-driven drug discovery part is also there. So you can see the video or like the presentation also. Now coming on how you can enroll. So just a second, the page is loading. Mm, it's still loading. Yeah, it. So as you can see that this is a program page, and what all I have just explained you. So you can see all that thing we have summarized in the program overview section, like what all the topics and concepts of what all access you will be getting. And apart from that, you will what all like resources you will be getting, like training, mentorship, then cloud tools and project support. And coming to the pricing part, as we can see, like for 45 days it is $350, and for 60 days it is $450, and for 90 days it is $550. And let's say for Indian community, you can see Indian or Asian community, you can see there's a drop down here. So just click on that thing, and you can like change your denomination or a currency from wherever you belonging to. So for Indian community, you can see the prices have changed and they have already been discounted. So for 45 days we have like 8,999. For 60 days we have 14,999, and for 90 days we have 19,999. And for like 90 days also we have something similar to this also. And let's say for like African community, they can also view the price. Let's say if you're from Nigeria, then you can see the prices here in the in Naira. So how to get yourself enrolled? So let's say I'm interested for the 90 days part. You can see all the resources which I have just mentioned you. You simply have to click on buy now, and yeah, you will be coming at this checkout page, and you can simply enter your user details. Don't forget to click on this checkbox, and after that you can select any of the method like Stripe or PayPal. Or also we have an alternative like methods for our Indian community via UPI or bank transfer, and for other participants also we have the alternative payment option using Payden. So this was all about the we can say the registration part. So if you have any queries, please feel free to ask. Thank you, everyone.
Thank you so much, Shubham. So we'll pause for a couple of seconds for the participants to share their queries. You can either unmute yourself and ask, or please feel free to post it in the chat, and we'll be happy to answer. We have one query from Professor Hussein. He's asking, can I get any discount as fellow?
Yes, Professor Dr. Hussain. Like we have several scholarships ranging from 20 to 50%. As you can see here on the screen, I think it is still visible. Yeah. So we have some scholarships ranging from 20 to 50%. So you can mail us your details for the scholarship part at marketing@omicslogic. I'll just share you the mail ID. You can mail us at this ID, and we will discuss with our team and provide you with a suitable scholarship part. Yeah, thank you, Shigori. She has already given her details to contact us. So you can email us at marketing@omicslogic.com, or you can also message us at this particular number. It is also available on WhatsApp. So both, like whichever part you think is feasible, you can message us.
Thank you so much, Shubham. And we have also shared the links to our social groups on WhatsApp and Telegram as well. So please feel free to join that group where you can post your queries as well. And with that, does anyone else have any further queries about joining the research fellowship program or about the program registration? If so, we'll be happy to answer. Okay, it looks like no one has no further queries. If so, we'll be ending today's webinar. All right, then thank you so much everyone for joining today's webinar. And if you have any queries, once again, please feel free to reach out to us through the email ID and the contact number that we have provided. And with that, thank you so much everyone. And we look forward to seeing you at the Research Fellowship Program, and have a great day. Thank you.